Dear Kavli Prize laureates, Prezes, Kavli Award Committee, ladies and gentlemen, it's a distinct pleasure to welcome you to campus and to the Oslo Science Park. So my name is Svein Stølen. I'm the rector of the University of Oslo. And I appreciate greatly that the Kavli Prize Award lectures take place at or close to our campus today, because the university was established at the 2nd of September 1811. So we are celebrating today. So dear laureates, your lectures today will clearly contribute immensely to our celebration, so thank you for that. Thank you so much for your dedication and energy, your will, and congratulations. I'm a strong believer in science, in basic science. This is the bedrock of research-intensive universities and the University of Oslo. At the same time, I believe that we can take our knowledge more efficiently into use faster than we have done in the past, at least faster than we've been able to do at the University of Oslo. Different people, different skills must meet, so we need seats that secure that different roads are crossing. Oslo Science Park and Oslo Science City is such a site where we cultivate curiosity, excellence, new ideas, but also a site where science meet across disciplinary boundaries and across sectors of society. Knowledge is power, but first and foremost, when new insights are translated into use to the benefit of mankind. Such is clearly the case for the stories to be told here today. Less traveled roads that meets and that make all the difference. Stories that must be told and explained to the society and to politicians, that showcase what science is, and why science matters. Stories that will contribute to continued support for science and research, also for blue sky science and research. So it gives me a great pleasure to welcome you. It gives me also great pleasure to give the word to Prezes, President of the Norwegian Academy of Science and Letters, Lise Øverås. Welcome.
Thank you. And dear Kavli Prize laureates, Rector, Kavli Foundation, Ministry, and Friends of Science. A warm welcome to all of you to this Kavli Prize laureate lecture in nanoscience at the Oslo Science Park. Please let me first say thank you, Rector Svein Stølen and the University of Oslo, and also Oslo Science Park, for your great cooperation and for hosting us in this wonderful venue here today. And let me also say congratulations to the University of Oslo. I really look forward to celebrate also the day with you. The Kavli Prize honors scientists for breakthroughs in astrophysics, nanoscience, and neuroscience, and thus transforming our understanding of the big, the small, and the most complex, to quote Fred Kavli himself. Since the first award in 2008, the Kavli Prize has recognized innovative scientific research from the discovery of CRISPR-Cas9 to the detection of gravitational waves. The Kavli Prizes have so far honored 65 scientists from 13 different countries. 10 laureates have also received the Nobel Prize after receiving the Kavli Prize. The Kavli Prize was established in 2008 by Fred Kavli, a Norwegian-American physicist, entrepreneur, and philanthropist who turned his lifelong fascination with science into a lasting legacy for recognizing scientific breakthroughs and for supporting research. The Kavli Prize is a cooperation between three partners: the Norwegian Academy of Science and Letters, who is administering the Kavli Prize, together with the Norwegian Ministry of Education and Research and the Kavli Foundation in the United States. It is paramount that the choice of laureates is recognized globally. The Norwegian Academy of Science and Letters appoints 3 Kavli Prize committees on the basis of nominations received by leading international academies and other equivalent scientific organizations. Each prize committee consists of 5 members who are all world-renowned scientists. The prize committees are tasked with reviewing and discussing the nomination received through an open international call and putting forth an unanimous recommendation for the laureates to the Norwegian Academy. So let me take this opportunity to say a big thank you to the Kavli Prize Nanoscience Committee led by Professor Bodil Holst. You have done a great job in identifying these three outstanding scientists, and we all sincerely appreciate your effort in this. This week, we call it the Kavli Prize Week 2024. We want to celebrate not only excellent science, but also put the focus on the scientists behind the science. The Kavli Prize Week includes symposiums, lectures, the awards ceremony, banquets in Oslo and Trondheim, and interviews with the laureates, so we can really get to know them better. But here and now, we look forward to the 2024 laureate lectures in nanoscience. So please let me introduce Naomi Halas, member of the Kavli Prize Nanoscience Committee. She will lead us through today's session. So Naomi, please take the floor, and thank you.
Thank you very much, Lisa. Yes, I'm Naomi Halas. I'm a member of the Nanoscience Committee. I'm a professor at Rice University in Houston, Texas. So it's been an honor to represent the Kavli Prize Committee, and I just want to actually acknowledge our wonderful committee members. So Bodil Holst was the chair of our committee from the University of Bergen, Norway. Daniel Esteve from Saclay in France, the French Alternative Energies and Atomic Energy Commission. Tanya Weil from the Max Planck Institute for Polymer Research in Germany, and Shuitong Li from Suzhou University. So they are all here with us today. So our work involved really going through a host of nominations submitted by scientists around the world, spirited discussions of potential candidates and possible themes, and after all of this work, our committee emerged with very strong and unanimous support for the topic of this year's Kavli Prize in Nanoscience, really for support of the nanoscience and exploring biological functions at the nanoscale. So this year's awardees, as you can see, this year's laureates are Robert Langer from Massachusetts Institute of Technology and the Koch Institute of Integrative Cancer Research, Armand Paul Alivisatos from the University of Chicago, and Chad Mirkin from Northwestern University. And our citation is for pioneering work integrating synthetic nanoscale materials with biological function for biomedical applications. So in the 1960s, there was a very thought-provoking science fiction film called Fantastic Voyage, and many of you might have remembered that film. This is where scientists were physically reduced to nanoscale dimensions to enter a person and save their life. So real nanoscience has enabled our laureates to do even better, okay? To create new nano-engineered materials, nanoscale devices and structures that can interact with cells, enter cells, probe and manipulate genetic material, and illuminate and guide the way to create new diagnostics, vaccines, and therapies that work at the intersection of nanoscience and life itself. These three laureates are truly the pioneers of our modern-day fantastic voyage. So now I would like to introduce our first laureate, Robert Langer from the Koch Institute of Integrative Cancer Research. And I like to think of in every field where there are pioneers, there's one true person who stands as the true first pioneer of a field. And I feel that that's very much describes Robert Langer in his contributions. I think of him, well, as an American, I was thinking maybe I should call him the Neil Armstrong of nanomedicine. But here, I think it would be much more appropriate to call him the Roald Amundsen of nanoscience and nanomedicine. So with that, I would like to hand the floor over to Bob Langer to hear his presentation today. Bob.
Thank you. Well, thank you so much. This is a tremendous honor for me, and also to share it with Chad and Paul, who've done just remarkable work. I thought I'd just tell you a little bit about how I got started. So I got my doctorate. I'm older than everybody else, so I got my doctorate in 1974 from MIT in chemical engineering. And when I got done that year, pretty much everyone in chemical engineering went to work for oil companies in the United States. So I thought that's what I'd do too. You know, in fact, if you had a car in 1974 in Boston, you literally had to wait for 2 hours at the gas station to get your tank filled. But the consequence of that for being, if you were a chemical engineer, is you got a lot of job offers. So I wasn't that good, but I got 20 job offers from oil companies, 4 from Exxon alone. And so, I thought I'd do what everybody else did. I'd work in an oil company. But as I went to these companies, I still remember one of my interviews at Exxon. I went to Exxon in Baton Rouge, Louisiana, and one of the older engineers said to me, if you could just increase the yield of this one petrochemical by like 0.1%, he said, that'd be fantastic. It'd be worth billions of dollars. But I remember flying back to Boston that night thinking to myself, I really didn't want to spend my life doing that. It just seemed kind of boring to, increase something by such a tiny percentage. And it made me think what I did want to do. And when I was a graduate student and I was supposed to be doing my PhD thesis, I actually spent a lot of time helping start a school for disadvantaged high school kids. And I got very involved in developing new chemistry and math curricula. And one day I saw an ad for City College of New York to be an assistant professor of chemistry to do just that. So I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea, so I found all the ads I could to do something like that. I found about 40 of them, and I wrote to all of them, and I don't think any of them wrote me back. I don't think I was in the right box, the chemical education box. So that wasn't going so well. So I started to think, well, how else could I use my chemical engineering background to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools. They didn't write me back either. But one day I was walking into the lab that I was in, and one of the older scientists said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman, and he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But Dr. Folkman had this idea, and I'll illustrate this on this slide, which might seem straightforward today, but it wasn't then. And his idea was that that a tumor, the reason it can be so bad is it secretes a signal, a chemical signal he called tumor angiogenesis factor. And normally you have your blood vessels, they're quiescent, they don't do anything. But his theory was that this signal caused blood vessels to start going like crazy to the tumor. And they feed the tumor, they give it the nutrition. And that enables the tumor to get bigger and bigger and bigger, and ultimately spread through those blood vessels. That's a process called metastasis, which eventually can kill. So his theory was that if you could stop blood vessels, that might be a whole new way of treating cancer. Well, I thought this was great. I didn't know anything about biology. I still probably don't. But at any rate, it turns out he was widely criticized for this in like different granting agencies and journals. But I thought it was wonderful. And he asked me, well, he said, your job, he said, would be to see if you could prove that I'm right, and in so doing, isolate the first blood vessel inhibitors. Now, one of the key challenges in that is you have to have a way to study blood vessels, and there really wasn't any at that time. So what we did is there's certain parts, organisms or parts of the body that really have almost no background blood vessels. The problem with developing the assay is most places do. But there's a couple places that don't, like the chick chorial and talc membrane and cornea. And so what we began doing was, this was the idea for the assay, that you'd have some type of tumor that you'd put in for the cancer specialists. These were V2 carcinomas. And then we'd have a little tiny particle, like a micro or nanoparticle, we could put next to it. And we needed these particles to be able to deliver over time large molecules that we were isolating. That was our theory, that some of those might be able to stop blood vessels. So actually, I was the only engineer in the entire hospital then. They didn't hire engineers very often. Most everybody was a surgeon or a biologist. So I actually spent a lot of time trying to do this. Dr. Folkman, actually, there was only one company in the world in 1974 really trying to work on advanced drug delivery systems. That was a company called ALZA. And Dr. Folkman was on their board, and they had very famous chemists on their board, like Paul Flory, who won a Nobel Prize, and he'd ask them, well, could they help us? But pretty much what everybody would tell him was that you really couldn't, because if you tried to put a large molecule, we were mostly focusing on polymers then, if you tried to put one inside a little polymer particle, it couldn't necessarily get out. Or if it could get out, it would go out immediately. So I spent 2 years experimenting with different techniques, different designs, different materials. I actually found over 200 different ways to get this to not work. But eventually, I was able to make tiny little microparticles or nanoparticles. This is just an early picture of one of them, and it's a picture, and here it is cut in half. And in 1976, we published in the journal Nature that you could use this for the first time to release these large molecules over time. It was also the very first time that nucleic acids were ever delivered from any particle. Nucleic acids, for reference, are things like DNA or RNA, which I'll come to later. So anyhow, we published that in 1976. As I'll go over, it was not very well received, but I thought I'd just show you a slide 47 years later, last year, where several references pointed out that this was actually the very first time, different Nature articles, and would even raise the possibility of using DNA or RNA as a drug. But let me go back to 1976. And when we first published this, I still remember giving a talk. There was a big material science meeting in actually Midland, Michigan. And I practiced my talk. I was 27 years old then. I practiced my talk for weeks into a tape recorder. And finally the day came, and I was, got up and I gave the talk. And I thought at the end of it, I didn't do that badly. I didn't forget too much what I meant to say, and I didn't stammer too much. So I thought when I was done with this talk, all these older chemists and engineers, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, we don't believe anything you just said. In particular, they said that what the ALSA people said, the molecules were too big, and they also said that the solvents we were using, which were things like methylene chloride or ethanol, they'd destroy whatever we put in. So that wasn't so good, but I kept at it. Nonetheless, one of the things when you're a young scientist you want to do is you want to get research grants. So I wrote research grants, and my first 9 were rejected. They were generally to places like the National Institutes of Health in the United States, and people would say, well, Dr. Langer is an engineer. He knows, I remember one of the cancer ones, he's an engineer. He knows nothing about biology and even less about oncology. I wasn't sure. I thought I knew enough about math to know that was pretty hard to do. But at any rate, I didn't get grants for a long time. And I actually loved being a postdoc at Children's Hospital. Like I said, I was the only engineer there, and I learned a tremendous amount. But a lot of my friends said it's probably not good to spend your entire life being a postdoc. I began writing for faculty positions, and like I said, I'm a chemical engineer. No chemical engineering department in the United States would hire me. They all said actually the opposite of what the funding agency said. They said, well, Dr. Langer wants to do bio things, and engineers, they don't do that experimentally. You should be doing energy or oil or something like that. So I got no jobs from any chemical engineering department. I finally got a job in a nutrition department. And the reason I think I got it is the man who was the head of that department, Nevin Scrimshaw, was a very famous nutritionist. He was also kind of a benevolent dictator kind of department head. So he liked me, so he offered me the job, but he didn't ask anybody else in the department what they thought. Which actually would have been okay except for one thing, which was that the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. And I'll back that up. This is actually from Michael Marletta, who's a very famous chemist. He was head of Berkeley's chemistry department, had a Scripps at one point. And he was giving, a couple years ago, I won some award, and he was giving a speech about me. I just excerpted this. They actually published it. And he was talking about what it was like to be with me in 1979. And he said one evening he went to a faculty dinner in a Chinese restaurant with me and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. Well, anyhow, that was not the best beginning of anybody's career. But I'm still there after all these years. And because we had done that work on the delivery systems, we did have a bioassay, and we could use that to, for the very first time, study blood vessel inhibitors. And so I did all kinds of that was the other part. I was isolating all kinds of things. And I had many different what I'll call fractions. They weren't purified. And I was testing them. And I want to show you the rabbit eye assay that we did. But if anybody's squeamish and doesn't like the sight of blood, you shouldn't look. But it's quite graphic what I'm going to show you. This is from a paper we published in Science in 1976. So like I say, don't look if you don't like. But what you see, and we did the one on the left, maybe over 1,000 times. We had, what I'll call a lot of controls because I had all these fractions that didn't work. And you always get what you see there. But with one fraction, we repeated it 20 times, you get what you see on the right. The vessels are lower, they're sparser, they avoid the tumor. So this showed for the first time that blood vessel inhibitors did exist, and it provided a pathway for future inhibitors to be studied. It took, medicine is, as I'm sure you'll hear from my colleagues, a very, very slow process in terms of moving from a discovery like that to real products. Here it took 28 years from that Science paper, using our assays and other things before the very first blood vessel inhibitor would get approved by FDA. And that took over $1 billion and really great work by Genentech and Napoleon Ferrar and others. But what you see, and this may be hard to see in the back, doesn't really matter, but what you'll see is starting at 2004, 28 years after that paper, the very first blood vessel inhibitor, Avastin, gets approved. Avastin, by the way, is probably the number 2 or 3 best-selling biotech drug in history. And what you see is many, this isn't even a complete list, but many blood vessel inhibitors are approved. The journal Nature estimates that 500 million people will be treated with these, and every year many, many millions are. Not only for cancer, but there are diseases of blindness in the back of the eye like macular degeneration and diabetic retinopathy. There were no pharmacologic treatments for this disease before these blood vessel inhibitors. But now you have drugs like Eylea and Lucentis, which are blockbusters, and again, sometimes even reverse blindness for these patients. So and you see they're even getting approved today, right? There's even, even in the last couple of years, there's many of these get approved, and that'll continue. I want to go back, though, to the 1970s. So this was what happened with the angiogenesis work. But in the 1970s, I kept working on the drug delivery aspects as well. And, one of the things that Dr. Folkman and I talked about was that maybe we should file a patent. Today, if people have followed what I've done, or Chad or Paul, lots of us have patents. In 1976, I had 0 patents. Boston Children's Hospital, where I was, which was a very good hospital, they had 0 patents, too. So we filed for a patent for the first time, and 5 years in a row, it got rejected. And I remember the head of the tech transfer office at the hospital coming to see me one day, and he said, "Bob, you are wasting so much money. The patent office is never going to allow this patent. The examiner doesn't like us or whatever." And he said, just please give up. You're wasting so much money. But, I don't like to give up. So I started to think, how could we convince the patent examiner to allow this patent, legally, of course. And science really wasn't working. But as I told you in the beginning, when I first started doing this, everyone said it was impossible, it could never work. I wondered, maybe somebody wrote that down. So, in 1982, I did this science citation search to see if anybody wrote about us. And it's always interesting to do that. So I actually found this paper, and actually there were others too, but this was, I thought, the best one, from 5 very well-known material scientists, and in 1979, describing this young field of drug delivery. I'll just read to you what they wrote. They said, generally, the agent to be released is a small molecule with a molecular weight no larger than a few hundred. One wouldn't expect that macromolecules could be released because of their extremely small permeation rates. However, myself and Folkman, we reported some surprising results that clearly demonstrate the opposite. So I really like that word, surprising. I thought maybe we could convince the examiner with that. So anyhow, the people at the hospital showed it to the examiner, and he said, gee, he had no idea about this. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits, written statements from each of these 5 people that they really wrote that. So it's a true story. So anyhow, I remember being a young professor, and I wrote each of these well-known people, and they were all nice enough to write me back that they really did write it. And we got this very broad patent. And that led to another thing. I had this dream when I started that something I did might someday help people. But, I realized even now working for 10 years, lots of people cited what we did in papers, but it wasn't leading to products. So finally, I start getting these calls from some giant pharmaceutical companies, one in multi-billion-dollar companies, one in animal health, one in human health. And could I consult for them on delivery of stuff like this? And what happened was I told them about the patent. They said, well, we want to license it and develop it. So what they did is they gave me a consulting fee. They gave me a grant for several $100,000. But most exciting to me is they were going to develop it. And they'd probably spent maybe a year each working on it, and then they just gave up. They didn't get exactly what they wanted. So, I remember being very discouraged about that. So one of my friends, Alex Slebanoff, said to me one day, Bob, he said, we should start our own company. So we did. We started a little company, and I got involved in starting many companies over the years. And those have led to all kinds of products based on these ideas. A lot of these are microspheres or implants that you inject or implant, but they've led to treatments for hundreds of millions of people, ranging from advanced prostate cancer, drug-eluting stents, even things in aquaculture, but all kinds of different opioid addiction and so forth. If you didn't have the delivery systems, those drugs often wouldn't work. They'd get destroyed or patients wouldn't take them. Now, we realized also that a lot of these were bigger, and if you wanted to deliver really small things, you could use these approaches, but sometimes the particles would aggregate, stick together. Sometimes they'd also be eaten by macrophages. So we identified with Vladimir Torchelin at Northeastern, 7 characteristics we want to build into nanoparticles, and we came up with another paper. This was a paper we wrote in Science, and that in a paper we wrote along with it a few years later on nanoparticles in medicine. They've been cited over collectively 13,000 times. But basically the idea we thought with PEG is that you could, which is, by the way, in Miralax, if anybody needs to go to the bathroom, you can take that too. But so it's been used for many years as a safe oral molecule. So basically we made these, and just to show that they're nanoparticles, we did atomic force microscopy, quasi-elastic light scattering, again, showing they're small. Most exciting, though, is if you put like an orange dye in these, and you ask, will the macrophages eat them? What you see is, without PEG, in an hour, you get a lot of ones eaten, but with PEG, you don't. So, so let me also show you that you could maybe even think about targeting them, by adding a certain ligand like an antibody or something else. Maybe you could target them to specific cells. I thought I'd put some of these things together and just show you a video that the TV show Nova once did on our work. And here it is.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should probably tell you that a lot of the clinicians I work with tell me it doesn't blow the cell up quite like that. But it gives you the idea. And this exciting thing about this is that you could not only deliver small molecules this way, but maybe opening the door, as that one article said, to delivering genetic therapies like siRNA or messenger RNA, things that could either turn genes on or off. So I thought I'd try to give an example of where this might be useful. So, and that is in gene medicine. In fact, if you talk to people, in fact, somebody even told me this last night, but it's been said by many people that the 3 problems in gene therapy are delivery, delivery, and delivery. And Phil Sharp, my colleague at MIT, who's a Nobel laureate, said, if you're going to deliver RNA or something like that, you need to solve the delivery problem. One of the things I got involved in, I'll get into this in a little more detail, starting other companies to try to address this. But from a science standpoint, one of the other things, you could use polymers, you could also use lipids. In the area of lipids, lipid nanoparticles have 4 components: cholesterol, a PEG lipid based in part on what I mentioned before, a structural lipid, and finally an ionizable, what cationic lipid. In 1987, there were several groups, Phil Feldner and Wu, pointing out that either with polymers or lipids, you could use cations to complex nucleic acids. But if you use cations, they might cause complement activation and other toxicity issues. They also wouldn't help you get out of what's called the endosome in a cell. So in the year 2000, Dan Pack and David Putnam in our group, we published a paper showing that you could make what we call ionizable cationic lipids or materials so they'd be neutral at basically pH 7. They don't cause a problem. But when they get to a lower pH, they become ionizable, and that helps you get out of the endosome. And then a Canadian group in Vancouver has done a lot of work on lipids. That's Semple et al., Peter Cullis's group, and many others over time. Now, the example I want to give you, to go into a little bit more detail, actually has to do with, could you come up with RNA therapeutics? So the idea that, you know, the central dogma of molecular biology is DNA makes RNA makes proteins. So protein therapeutics by 2010, there's so many, I mean, 70% of the top drugs are proteins, but it takes a tremendously long time to make them. Just to pick one example, if you have flu vaccine, you have to start a couple years in advance guessing what it's going to be, H1N1, H1N2, so that's why it's only 40% effective. And there are many other issues with protein therapeutics. Still, it's been revolutionary. Could you make therapeutics out of DNA or RNA? Well, out of RNA, you know, there's a classic papers around 1990 where people started, Wolf's paper in Science, where showing you that you could use RNA in animals, but it would cause some safety issues, very serious ones. So in 2005, Carrico and Weissman showed that by chemically modifying the RNA, you could avoid those problems, and it would be quite safe. They want to start a company, and so they had a patent. They tried to start a company out of Penn. Nobody would give them money. They all said, how are you going to solve the delivery problem? How are we ever going to get this to work? So they never were able to do it. About 5 years later, Derek Grossi, a biologist at Harvard, had actually also used this kind of approach. And he wanted to start a company, and he talked to Third Rock and other good venture capitalists, and they all said, well, you'll never be able to do that. He came over to see me one day, and I said, well, I actually think we can solve these delivery problems. You know, why did I think that? Well, first, we'd shown back with that Nature paper in '76 that you could deliver large molecules. We'd further showed the idea about PEG, that we could do ionizable components. We had also showed, which I didn't talk about, work that Dan Anderson in our lab had done, making literally thousands of lipids, so you'd have all kinds of things to choose from, and work that Rohit Karnik had done in our lab using microfluidics to actually manufacture nanoparticles. Again, all of it was very basic work. You're not going to ever get this without having a company. But that being said, you know, I told Derek, well, let's talk to Nubar Fayan, who I was on his thesis committee at MIT, and he had started Flagship Pioneering, and we also got Ken Chen, who was a cardiologist involved, and we started this company. We called it Moderna. And the idea, like I say, is we put messenger RNA in little nanoparticles. And not only could you theoretically make any protein, you could also even make proteins in areas that people had never looked at before, like intracellular ones or membrane-bound ones. And Moderna, I can tell you, all these stock analysts and many, many clinicians and scientists said Moderna is a joke, it'll never work, it'll be the next Theranos and so forth. But the scientists at Moderna, a number of whom are my students, you know, they kept working on this, and by 2019, in spite of all that criticism, we had 13 products in clinical trials, early stage, and then, as you all know, COVID hit. I want to show you what the Moderna scientists were able to do, which was pretty unprecedented. In January 11th, 2000, the Chinese scientists published the virus genetic sequence. Took less than 2 days to design this. Put it in the nanoparticles, ship it to our clinical partner, NIH, for testing. First patients were treated in March, and by the end of the year, we had done over 30,000 patients. And this would then get approved by the FDA for emergency use authorization. Just to show you some data. if you look at vaccinations, unvaccinated with no booster or with a booster, you see the enormous differences. If you look at effectiveness, this is just in the United States. In fact, only 2 up till recently just got approved by FDA, but Moderna was 94%, Pfizer and BioNTech 91%, and Johnson & Johnson had a adenoviral approach, but would ultimately get pulled from the market. This is a very recent study in JAMA, again showing you that Moderna is the safest and most effective vaccine. You know, one of the things that comes up to some people is, well, okay, it's effective. It's, you know, flu might be 40% effective. This is much greater, but is it safe? And I think such an important thing to remember is no medicine is completely safe. And no vaccine, even though those are generally considered the safest and best treatments for anybody if you can do it, are completely safe. So maybe a comparison is people have had flu for years. Are there what's more safer, the mRNA vaccines for COVID or for flu? And here you see one of many examples showing that I'd say the mRNA vaccines are slightly safer. If you look at other analyses, you know, you look at the enormous number of failures. A lot of times now people say, well, couldn't anybody do it? Well, the fact is, if you look in March 2016, there are 49 failed vaccines, 2 approved. And you can see the same thing in treatments and antivirals. And also, I think economically, it's prevented over 3 million deaths, prevented over 18 million hospitalizations, almost 120 million infections, and saved the US at least over $1 trillion. And this, to me, is just the tip of the iceberg. There are many, I think Moderna and BioNTech and others now, all kinds of large pharmaceutical companies are doing this. You know, but one of the exciting things to me is cancer. Could you come up with new ways of treating cancer? And one of the things, this is from Moderna and Merck, is here's 157 patients with advanced melanoma. And what they're doing, it's really analogous to the COVID vaccine, but now you select 34 mutations known as neoepitopes in the patient's cancer cells, and you incorporate the genetic code in those neoepitopes in exactly the same way it was done for the COVID vaccine, mRNA and nanoparticles. And the best treatment in the world right now is Merck's drug Keytruda. That's an immunotherapy drug. So here what's done is half get Keytruda plus the personalized cancer vaccine, half gets Keytruda. But the results are, at 2 years, reduction of recurrence or death is 44%, 3 years, 49%. Also, the reduction of metastasis is over 60%. So I thought I would end a little bit by where we're going in the future. A couple years ago, well, actually 12 years ago, I had a visitor who was a software expert, and he came to see me, and he was very concerned about vaccines too. This was way before COVID. Here he is, and me, that's me on the left, and that's the software person on the right. It's Bill Gates if you don't recognize him. And one of the things that he said is, you know, people in the developing world, this was 2012, don't take their vaccines. He said, could you someday come up with a way that you could give what we'll call self-boosting vaccines, just one injection, and you'd get pulses like you see here. And what Anna Jakulenik and others in our lab did is we made tiny little particles, like little tiny eggshells. But by changing the polymer chemistry, we could make them break at almost any time, not just time 0, but 1 month, 2 months, whatever. And you do that by changing the molecular weight of the shell. Here we're using a degradable material, that polylactic glycolic acid, that breaks down into water and CO2. But she did that, and these are just some pictures of these. Here, the blue dye represents the vaccine. But here you can see that you can actually do this. This is one animal, and it gets 11 different pulses at different times. And where we hope we take it someday, and again, we're not there yet, is we've also developed all kinds of microneedle patches that you could use. They're like little Band-Aids. And if you look at an SEM of it, what you see is they don't go very deep, so they don't cause much pain, but they could deliver these. And you could basically, we're working with Mark Prausnitz, who's here, who's one of my former students now at Georgia Tech. Here's a little patch, and you could hopefully ship these all over the world if you can stabilize them, which we've also been working on, and you know, just put them on people. So those are some of the things that we're trying to do. And so I just want to end by saying that it's a really tremendous honor for me to be here. I think you're going to hear two fantastic talks right after this on other aspects of biomedicine and nanomedicine. But I really believe that the kind of work that the three of us are doing, and of course many people all over the world in nanomedicine, we're really at the earliest stages of what it can do. I hope this is one example. You'll see two more really good ones right now. But I really feel it has the opportunity to have a tremendous effect on relieving suffering and prolonging life. Thank you so much.
Thank you so much, Bob. Do we have any questions? Well, I've got a question.
Sure.
You mentioned the many companies that you started, and you also mentioned some of your former students who are now professors. Do you have a tally? Do you know how many of your former students? I know we have some at Rice, of course,
Sure.
several, and I worked with one actually years ago.
Well, you have three at Rice, yeah.
Okay, so can you get just in a description of your impact academically? Do you have a count of how many of your students are in professorships now?
Yeah. Well, there's over 400 that are professors. I mean, and they're everywhere. They're at the University of Chicago. They're at Northwestern. I think actually, by last count, I think 10 are at MIT, 11 at Harvard, 12 at Stanford. But they're all over the world, and so that means a great deal to me that they've done so well. I think that's the other part of being a professor, right? You try to do this kind of research, you teach, and hopefully you train people that will go on and do far better than we do ourselves.
Yes, and also we're here at this wonderful science park. And this is also a very important aspect of nanoscience that we don't see with astrophysics and we don't see yet with neuroscience. But the entrepreneurship side, being able to get things out into society is very large, is really very huge. And you've obviously convinced us this is extremely important. Do you have any suggestions or any advice for the entrepreneurs who are people who are thinking about this? This landscape is different in Europe than it is in the U.S. Is there something that you could suggest that people facilitate here, or any advice you could give?
Sure. Well, actually, all three of us have done things with companies, and I guess, there are a lot of pieces of advice. I'm not sure where to start. One thing, though, that I found is certainly very important, which I mentioned, are patents. Different people have criticized patents, but the fact is, especially in medicine, if you don't have a patent, you'll almost never get the type of funding you need to create these kinds of things, especially if it's something that's going to be a medical treatment. And even in the diagnostic area, I'm sure Chad and Paul can go over that, it takes a lot of money there too. It's not the same thing as like Facebook or something else. It takes a tremendous investment. So what happens is, you can get some money from the government to get us started, and that's wonderful. The grants, because so much basic research is done at the university, and without that, these things wouldn't happen. But it also wouldn't happen without investments. So I'd make a couple of points on that. First, patents are important because investors, rich people or venture funds, they won't invest in something if they feel after putting in $1 billion, somebody else can do the same thing for free. Just won't happen. I've seen that over and over again. Really smart people asking me, they've published stuff in Nature or Science, and they said, well, we can't get any money. And I said, well, why do you? I said, I think you should. And he said, well, the problem is everybody tells us that we published it, and now nobody will patent it. What we do, by the way, is we do both. We publish everything and we patent everything. But the patents are important. I also think nations can do things that can, so the scientists can do patents. I think that what the nation can do, one of the things that's been, I think, very important in the U.S. are things like capital gains tax. Like if somebody does make an investment, they pay money and they sell their stock much later, well, they can make more money than just getting like a salary or something like that. And so I think that's important. I also think that to me, whenever you do these things, it's a team effort. I might be able to help on the science, but you need, clinicians for clinical trials and clinical trial design. You need manufacturing people. You need great business people. And so you really have to, if you're ever going to succeed, I think you really need a great team. And so I think somehow getting that team together is really, and working together is really important.
Very good. Any questions from folks here? So, well, I'm sure we're going to have some great discussions afterwards. So why don't we thank our speaker again.
Thank you.
Thank you so much,
Thanks so
Bob.
much.
Thank you so much, Bob. So our second speaker, so before we get started on our second speaker, I want to mention that in the world of biology, so we're not so many biologists here, so I can probably speak relatively freely. The icon that we use to symbolize biology and to symbolize medicine, it's not a cell, it's not DNA, it's actually a microscope. Because a microscope, more than any other instrument, is what really opened the door for our understanding of even the discovery of cells, the discovery of subcellular organelles, the discovery of so many important things. And so imaging and visualization is something that has been absolutely vital for the development and for our understanding of medicine. And so nanomedicine has brought us, or nanoscience has brought some very, very important tools. And in particular, our next laureate, Paul Alivisatos, is very much symbolic of really what you can do to visualize structures using new tools from nanoscience. So with that, I would like to introduce Paul Alivisatos from the University of Chicago to speak with us about nanocrystal building blocks that optimally produce and harvest light.
Yeah, thank you so much. So you just heard Bob Langer describe a young Bob Langer being in the wrong place sometimes, but finding ways to escape. I'm sort of hoping that will apply to me, because following Bob Langer, that's a pretty tough thing to do. But what an honor it is to be honored with Bob and with Chad, who are just amazing scientists. And so I'll tell a little bit about our journey in quantum dot biolabels and give you a feeling for that. So the three things I'll talk about are the idea of taking the artificial atoms, which were developed over a couple of decades, into the first use as biolabels. A little bit about taking those into larger assemblies that then can be used to detect things. And finally, what I hope will happen in the next 10 years, I believe it will become possible to control nanocrystals at the level of single atoms in due course, which is certainly something we don't quite know how to do yet. So the early part of my independent science career was spent studying very tiny crystals. So these are materials that might have hundreds or thousands of atoms in them, and it derived from my period as a postdoc with Louis Bruce, who along with Munji and Alexei were awarded the Nobel Prize for the study of quantum dots just a few months ago. So I was one of the very early scientists studying these tiny crystals that are somewhere between bulk solids and atoms. And in my near-death experience as a scientist, to echo some of Bob's, I spent some of my early time as an assistant professor studying the structure and phase transitions in nanoparticles, including things like why they melt at much lower temperatures and how that works, or the presence of defects and whether it was necessarily true that there would or would not be defects. For example, when we were first studying quantum dots at Bell Labs, I once went to speak to one of the most senior physical condensed matter theorists and showed him our early results with nanoparticles. And he looked at them for a little while and then said, you know, you seem like a very nice young man. My recommendation would be that you study something else, because you will never be able to make high quality crystals in liquid solutions. We all know you use vacuum chambers and evaporation techniques to produce things that are atomically perfect. So I spent some time trying to prove that wrong by studying whether defects had to be present in nanocrystals or not. And ultimately, it turns out that just like smaller diamonds tend to be more perfect than larger ones, in fact, it's much easier to make tiny crystals perfect than it is to make large ones perfect. So it was, in fact, the exact opposite, as is often the case. So I spent a lot of time doing that. And in fact, like Bob, I also had a near-death science experience in the sense that during the time that we were doing this work, I was never given any grants because people didn't see the point of it. And there were a lot of very unfriendly, or what seemed to be friendly, but really weren't very friendly reviews. What just happened here? We just lost our screen. Oh, there we go. So, but in any case, it did turn out that around the time that we were publishing these early papers, I went bankrupt at Berkeley, in fact, and they bailed me out. And they also gave me tenure because they were reading the papers, and they said, sooner or later, this is going to come to something. And in fact, the something that they came to was that, in the, let's say, mid-1990s, finally, when we were able to make these tiny crystals, they were very high-quality crystals. We also were able to harness the quantum size effect to do biological imaging. And that was a kind of breakthrough for the study of these materials because it was one of their first applications. I'll say a bit about the quantum size effect in a moment. But what I do want to emphasize is that the applications that we'll show were all ones that followed a period of time in which we spent a lot of effort really understanding the fundamental underlying chemistry and physics of these materials. And it was through that study that we were then in a position to take these materials and find their early uses in the quantum dot biolabeling. The other thing I'd like to point out, Bob showed very beautifully how various large organic macromolecules that are artificial can be combined with living, with macromolecules from nature. They can be combined and see great effects from that. And the question in this research will be, can we take inorganic tiny crystals and combine them with, say, proteins or DNA, as Chad will be showing. And the point is that by looking at very tiny crystals, they become the same size as the macromolecules that function so much throughout life as proteins and other components. They are just about exactly the same size. And as a consequence, the possibility of conjugating them together or being able to take these small solid-state materials which we're used to building electronics out of or other things, suddenly we can imagine that those things appear inside a biological system and help it to function. In our case, we'll be mostly looking at using it to illuminate, to see things, but ultimately what is down the road is the possibility of essentially having inorganic prostheses at the molecular level inside cell membranes and other things of that kind. That's where these things may eventually go. So why would these be interesting why would these tiny crystals be interesting in order to illuminate things inside cells? And the reason is because they have luminescence spectra that can span an enormous range in the visible spectrum. And to explain the quantum size effect just briefly, most of you will probably be familiar with it, but the analogy that I like to use is the one that if you go to a science museum, you'll often see one of those parabolic funnels, which if you drop a ball into it, the ball will go around, and then as it comes down towards the center, it will spin faster and faster. And a quantum dot's a little bit like that. If you imagine the electrons holding all the atoms together in the crystal, and you put in enough energy to knock an electron out of its bound state, it will start to zip around in that crystal. And the smaller the crystal, the faster that will happen. As a consequence, when the system now relaxes, that high energy electron falls back into the hole that's been left behind. The energy of that photon that's emitted will be a higher energy as the crystal becomes smaller. That's called the quantum size effect. And here you can see an example. And I think when this was published, it was pretty much the state of the art. You can do better today for sure. But at that time, the ability to make spectra that went all the way from the near infrared into the very heart of the visible was quite important because it meant that you could have a set of labels that could be used to visualize biological systems. And we'll see a little bit more about why that was important. First of all, dyes are typically what's used. In fact, Bob showed an example of some labeling with dyes before. And typically, dyes have some of the following features. They have in them, let's say a dye molecule might consist of 30, 40, 50 atoms, something in that scale at most. And they'll have discrete spectra of excitation and emission. And if you have a very small molecule like that, I'm calling it small, some people may think of it as large. In any case, if you've got, let's say, 20 atoms, and you make an electronic excitation inside of it, then it can relax back and emit light. But when that happens, actually moving one electron inside a system of that size changes the shape of the molecule. So it results in kind of reorganization. And as a consequence, there's built into the emission or into the excitation this kind of long tail that represents the reorganization at some level of the molecule inside it. So there's an inherent width to these that comes from the perturbation of the molecule when it's excited or de-excited. In the quantum dots, really the width of the emission would be quite a bit narrower than this if we had a single particle, and we'll show some of that soon. But this is really determined by the distribution of sizes. However, a difference for the quantum dot is that once you go past the excitation energy, there are just many, many, many energy levels that pile up. And so it tends to absorb very, very strongly in the blue and to emit very beautifully. And you could imagine here these two sizes, for example, if you had a single light source, you could excite both of them directly. So those are some of the things that are important. We'll talk about the photo bleaching in a moment. So once these quantum dots had been grown and were available, typically we coated them with an organic layer on the surface. It's shown here as a core shell. There's some details that matter in terms of making them bright emitters that I won't go into here. But prior to our work, they had, let's say, they looked something like the surface of a micelle. There would be a polar group here towards the nanoparticle surface, and then a non-polar group coating it. So we needed to make them compatible with going into biological specimens in order to use these light emitters in biology, and we did that by borrowing the well-known chemistry of silica. People had already for a very long time known how to take biological macromolecules and attach them to glass surfaces. So that was a very well-established chemistry, which we simply borrowed and then used a variety of ways to conjugate antibodies or to use the biotin-streptavidin interactions to take these silica-coated nanoparticles, and then they were available subsequently for directing to a specific spot inside a cell. So this was our first example of a biolabeling experiment where we directed quantum dots onto actin fibers or into locations in the nucleus of cells in order to be able to just simply see that we could do a two-color bioimaging experiment with dots. I do want to point out that our earliest paper was with my student Marcel Boucher, who went on to a really beautiful career in this field. He was a professor at Carnegie. He was part of the team that founded Quantum Dot Corporation, but then a few years later went to Carnegie Mellon. Sadly, Marcel passed away just 2 years ago at a very young age. And of course, the work was done with Shimon Weiss, my colleague from Lawrence Berkeley National Lab. And also, Shuming Ni published a very beautiful paper that was essentially back-to-back with ours that had some related ideas inside of it. So these biolabels, we'll see for a variety of reasons why they're so advantageous. They have many characteristics that make them really very favorable with respect to the use of dyes compared to those. And we decided in my research group very early on that we were not going to be the ones who would know the biological applications in full. And therefore, we just felt an imperative to commercialize them. So we did, as we were doing the earliest work before any publications, we did we sought extensive patents for this idea, which were ultimately awarded. And we went into, more or less simultaneously with the publication, we had already founded the company, which about 18 months later had its first commercial products on the marketplace. And that was really important to me, at least, that we would do that, because I had this deep sense my lab was not going to be the place that was going to do this. In fact, at that time, we had the experience that we were able to make the dots and do all of these functionalizations, and people would come to me whenever we did present these kinds of results and say, how did you do this? How did you do that? And so on. And we would always I would always invite them to come to my lab. And so we had at any given time dozens of visitors wanting to learn how to do these things in the laboratory, and it just became really obvious that we could not manage that flow anymore. We were completely overwhelmed by it. So again, it made complete sense to me that we would go forward and commercialize these things. This is an example of some of the very first quantum dot products, and this is an example of all being excited with one laser, but being able to make all the colors by that method. Now, let me talk about the photo bleaching issue because it's pretty fundamental, and I think you can see here, if you just look side by side, that the dye will bleach completely during the time that I'm speaking, whereas the quantum dots really will remain quite photostable, actually for months, as you'll see momentarily. And why is that? Well, think of it like this. As I mentioned to you, if we kick an electron out in that semiconductor, it kind of spins around the whole crystal, and that's the whole basis of the quantum size effect. What that means is you have one electronic excitation. Let's say it's a couple of electron volts worth of excess energy, 2, 3 electron volts worth of excess energy, distributed over many thousands of atoms. But in the dye molecule, you have that same amount of energy distributed over maybe 20 atoms. And now you think about, well, what can go wrong? Well, most of the time, these things will absorb that, and a photon will come back out, and it'll just do that over and over again. But every so often, just statistically, that energy will go someplace inside that molecule and undergo a chemical reaction that's unwanted. It'll be an oxidation event. It'll be the production of some kind of a photo destruction of the molecule. And just statistically, if you just want to think of it that way, when you have a quantum dot with, let's say, 5,000 atoms in it, compared to a dye with 20, there you go. You're going to have an enormous difference in the photostability. In fact, in many ways, quantum dots can be thought of as an object that can absorb light over a certain volume and then direct it to a particular location, but to do so in a way that protects the energy and doesn't allow it to go into unwanted channels. And that turns out to be, a pretty significant thing. The other thing to recall is that it will turn out that because the entire dot is absorbing light at a very high energy compared to where it emits, it can pile up a lot of oscillator strength, if you like. So the absorbance of a dot well above the band gap of the semiconductor can be about 100 times or even 1,000 times that of the strongest dye molecule. So they're really bright objects. And in fact, so that allows them to be used for a variety of things. So I'm just going to show very quickly a few examples of some of the things that we explored in the early days. Today, they're being used all over the place by thousands of scientists every year, probably about 10,000 research groups a year, one way or another, are using quantum dots for their biolabeling. But these are some of the things that we explored early on. Here's one that I still kind of like. If you just put the dots down on a surface and cells crawl over them, it turns out it's a funny thing. When cells crawl, and I had no idea this was true, but when cells crawl, they kind of ingest the stuff that they crawl over. And so they'll leave tracks behind, and you can observe the tracks. And it turns out that if you change the chemical environment of the cells, you can pick up immediately and all kinds of details about how that works. That's an example. There's lots of examples of dots being used as single dots. This is one from Maxime Dehaan, who was a postdoc working early on with Shimon Weiss and myself. And this is an example of looking at kinesin trajectories of single particles. And again, these are so bright, it's very easy to see a single particle quantum dot and to track its motion and so on. So they can be used to see things at the single dot level. Here's another example of that, in which a whole variety of things like float on membranes. You can either see ones which have very directed motions. You can see ones which have a kind of regular diffusive motions. You can see ones that are attached to rafts and therefore move in a much more confined way. And all of that can be imaged very readily without photo bleaching. This is one of my favorite things, even though I don't know how many biologists truly care about it, but I like this one for the reason that if you load dots into a cell, into a healthy cell, and you can load a whole bunch in there, load a certain amount, then the quantum dots will be passed to daughter cells. And so in this case, you can see an example where there are some breast cells. And as many of you may know, that if you take a three-dimensional tissue culture. Today people will often study organoids. If you take a three-dimensional tissue culture, the cells will start to divide. These breast cells will make a kind of so-called acinus, which is a kind of proto-breast gland with a center that's hollow, and around it all the cells. So this is an example of an experiment where the same quantum dots are probed over 3 months, and they don't bleach, and you can see which cells belong to which. So in this case, all the green labeled cells are benign breast cells, which divide and make this kind of proto-gland. And then introduced into the medium are various cancer cells, which proceed to go into that gel and seek out, through chemotaxis, they go directly towards those little breast glands, and they touch it. And then what the breast cells will do in the functioning situation is induce apoptosis in the cancerous cells and kill them. And so you can follow all of this for months. And this is an example in which the time tracking shows all the dying tumor cells surrounding that were observed over a period of 3 months. That shows that, that's something that you just can't do by other means, but by the luminescence, suddenly this kind of thing is observable. There's a lot of work that's being done to do this kind of tracking inside animals. This is an example from Shimon and company that shows the study of a prostate cancer, a mouse that's inflicted, that has unfortunately got a prostate cancer, and directing of the quantum dots to that location through the correct antibodies, but also observations in the sentinel lymph nodes and so on. This is a complicated topic because it has to do with introducing these inorganic materials into living species and so on. I mean, I personally think, what do I know, but my sense of it is that the kinds of applications in fundamental cell biology, like what I showed you about the cell divisions, or the ability to track phenomena inside cell, to understand fundamental cell biology, that's where quantum dots really shine at the moment. Our ability to put engineered nanoparticles into living systems in functional ways still has a long way to go, but it will come over time. I do want to take a little interlude here and just say that during this period of, let's say, the late 1990s to the early 2000s, for me it became, and I described a little bit, what happened around the formation of Quantum Dot Corporation, but for me a little bit, it also just became a kind of passion that for nanoscience to thrive, it was going to be important that we find new ways of sharing. And so we developed, of course, the company, the journal, which was a project with my spouse, Nicole Alivisatos, who those of you who published in Nano Letters in the early days will know. But also we created the Molecular Foundry. And the Molecular Foundry is a national user facility in the United States. It's a global facility. And in it, there are, at any given time, a few hundred scientists whose job is to be at the cutting edge of nanoscience, but to share what they do. And to me, that became an important part of how we would approach all of our different projects. In other words, we would constantly be trying to develop new materials, but as we developed them, we'd either get them into the marketplace, we'd certainly make sure we had a place to publish things rapidly, or in many cases, because they weren't ready for that yet, we would have a place where the methods for making those things were shared with everybody so that people could bring the best expertise to use. This also happened to be a period, and I don't think it's a coincidence, this sharing philosophy led to what I'll consider kind of like a Cambrian explosion of the diversity of nanoscience building blocks. All of a sudden, a huge number of different ways of making nanomaterials like these proliferated to a degree that you almost can't imagine how big it all was. And it encompassed materials from completely different approaches, all the way from the scanning probe techniques to the synthesis of inorganic and organic nanomaterials to the use of all of the molecular building blocks of life. All of those things, as well as microelectronics and so on, 2D materials, you name it, they all kind of converged. In fact, we had our own kind of Cambridge explosion And it was to go from making just these simple nanoparticles to learning how to make all of these other kinds of structures. We learned how to make branched nanoparticles, wires, striped particles, hollow particles, nested particles. By the way, Bob, we'd love some time to work with you to take an inorganic nanoparticle with some drug molecules in there and diffuse them out. But we also could make things that look like PD and F orbitals and so on. You know, it just went crazy for a little while. And so that led us to be thinking at the time about the idea that if we think of this as an artificial atom, then these kinds of things can become artificial molecules of different kinds, because there are many inorganic components connected together in 3 dimensions with all kinds of complexities. And that led us in turn also around the same period and around the same time as Chad to think about the idea of using the programmability of DNA in order to build artificial macromolecules. So we happened to have these papers back to each other in 1996 about the use of DNA to pattern things. And Chad's idea was much more practical, and he'll talk about it, and it worked. Our idea was a little bit more chaotic, and it makes beautiful images, but it didn't reach the kind of technology level that Chad has shown. Specifically, our idea was not to make arrays, but to attach exact numbers of DNAs to each particle, and then to use the DNA in order to program little nanocrystal molecules like these little Mickey Mouses that you see here, where we've made trimers of gold particles and so on. And this became a little cottage industry for us for a while. This is, for example, quantum dots. You can barely see the quantum dot in each case. There's a quantum dot, but there are 4 gold particles around them, or 3, or 2, or 1, and so on. So we could build what I would call nanocrystal molecules like that. And these turn out to be quite interesting because, for example, the gold particles, depending on their distance, will change the color of the spectrum, and you can detect that very readily. Here's an example of that. If we have one of these gold nanoparticle dimers joined by DNA, it's only 2 particles now. If Echo R5 enzyme comes and binds to it, there's a question. Does it just cleave right away, or does it bend the DNA first, bringing the particles closer together, and then cut it? That's okay. I'll wrap shortly. So here's an example of that. I hope this movie plays. We'll see. Here's an example of that where we're able to be cutting, and you can see that's an example of a specific spectrum changing. And it turns out, if you look at this, this little plateau here, if you do thousands and thousands of these, you'll find that that, in fact, is the enzyme bending the DNA. So there's ways to see all those things. Okay, 2 quick things about the future, and then we'll wrap. I'm very excited today because about, it's getting to be quite a few years now. I guess 2008 was our first paper where we first started to look at nanoparticles inside a liquid cell with the electron microscope. So now suddenly we have a tool, and this is my favorite tool of all the years that I've been working in this field, which is a number of decades now, because in the liquid cell electron microscope, particularly the graphene liquid cell, but also the silicon nitride-based ones, we can do all of these things. We can look at the forces between particles, we can watch them grow, we can watch them disappear, we can watch them rotate and obtain the structure. It takes a little while to run, and observe every single atom inside a nanoparticle. We can even observe those little DNA groupings floating around in solution and moving about and so on. And I believe the liquid cell will be the tool for us to ultimately gain atomic control over every atom inside a nanoparticle. It's going to happen. And what would happen if we did that is I believe we'd learn how to place nanoparticles much more precisely than we can today. And so one of the challenges out there is, rather than just tagging nanoparticles to locations, is to begin to embed them inside biological membranes or inside other components inside biological systems, so that the inorganic macromolecules embed inside the macromolecules of life with an organized structure. For example, in this case, imagining detecting electric fields near a voltage-gated ion channel, or in another case, being able to build a photosynthetic membrane. And I know this is something our chairperson is working on, to make artificial photosynthesis, and I believe that in due course will happen as well. I want to also acknowledge that in my life, and Naomi didn't quite mention it, but for any number of reasons, that have to do with the philosophy that I brought to sharing our nanoscience through companies and the molecular foundry and so on. I also found myself getting involved at different levels of being, let's say, director of Lawrence Berkeley Lab, or the provost at Berkeley, or now the president at the University of Chicago. And what I want to say that isn't often said in these kinds of things is that these are very special places where thousands of people are trying to make discoveries. And they're very generative. I feel like everything that I just described would have been, never could have happened without being here. You're celebrating, 200 years, 211 years of the University of Oslo. It's really quite important to celebrate these places of discovery. The discoveries we honor are special, but the places are the ones that actually build them as much. It's the community of people that builds them, not just single persons. Along those lines, I want to thank my many students and postdocs. This is an example. Only about a third of the people could come, and this was my surprise birthday party. But you get a feel here for lots of people have been involved in doing the work that I just described. So I'd like to thank all of them, my many mentors, and of course Nicole, and thank you for your attention.
Thank you, Paul, for that wonderful, really wonderful display of how much quantum dots can do for us. Do we have any questions from the audience? Yes. Oh, hold on a second. We're going to record this. So.
Can your quantum dot do the clean energy to save the planet?
Yeah. Yeah, that's another lecture. I don't need there is there's a lot of work in which nanoparticles are used in a variety of ways for energy transductions, and it turns out that on the nanoscale, not only can you achieve cost scalings. I mean, one thing I didn't describe so much here because it's less important for biology, but it turns out that being able to make an electronic quality material with the same technology that we use to make bulk chemicals lowers costs, allows large volume with high precision. This is one of the amazing things about nanocrystals in general, is that they can be every bit as high quality as the most sophisticated component in a computer chip, but they're made the way bulk chemicals are. So they change the cost structure. And also, it does turn out that any energy conversion processes, when you look at them on very tiny length scales, they don't operate exactly the same way. And they open up the possibilities of doing many important things like thermal diodes or heat diodes and things like that. Many phenomena occur in the nanoscale that change how energy will be harvested. So it's a whole topic, not today's topic. Yeah.
Any more questions? Yes.
Yeah, so you said they're beautiful talk and work, obviously. You said there are a few barriers to get to the next step. What are those and what do you need to do?
So right now, apologies, Chad, but we think of nanoparticles and we functionalize them. I believe at some point we'll be able to have strongly shaped nanoparticles where each facet has different functionality on it, and where their integration into a biological system is not just random, but is ordered with respect to the structure of the nanoparticle, and it's deliberate with respect to how it's embedded inside the biological system. That would produce a higher level of functionality that could emerge, because as you know, inside biological systems, everything is directional, and we can emulate that, and I think that will be a big advance, that at that point we'll be able to do a lot more with these systems. Yeah.
So I have a question also, really maybe perhaps this is for everyone, for all three of our laureates. But here we are in nanoscience, and we know that there's parallels in neuroscience. Have you seen any work in quantum dots moving into neuro at any level, neuro or brain? Can you comment a little bit on sort of the future of probing neurotransmitters or something like that?
Well, it's funny that you should ask that, because I can't remember how many years now that it's been since the Brain Initiative started, in the United States. But its origin was the Kavli Foundation coming to a group of us that I happened to be a part of and saying, let's have a workshop between nanoscientists and neuroscientists and see if there's a connection point. And everybody came to this conference. It was in the UK. There were, I think, 12 of us there, 6 and 6. And each of us had a chance to talk about our ideas, and none of us had any ideas. But then we spent a couple of days together, and before you knew it, it was obvious that there were lots of interconnections between the two, and there are many, many, many. I didn't talk about any such connections here, but fundamentally, designing different ways of imaging and collecting signals from the brain at large scale is one of the most important problems for decoding how the brain works. And that's emerging systematically in remarkable ways. That is a kind of convergence of neuroscience and nanoscience. So there's a very rich connection between the fields. So I think there's great prospects for it. And the Kavli Foundation was just spectacular during that period. They completely changed the global conversation about that.
Wonderful. Well, thank you so much.
Thank you.
So let's thank you. Very good. So, if we think a bit about the 20th century, right? So we can think of, what were the big breakthroughs in the 20th century? So we could argue, but I would say that there are maybe, 3. Computer is one. Being able to enter the atom and understand and harness atomic energy is the second one. And the third one is DNA. Right? So much has happened since the discovery of DNA and the DNA structure, but when it comes to nanoscience, what's so interesting is that DNA is no longer just DNA. If you attach DNA and integrate DNA with inorganic systems, it changes. The properties change. And I think our next laureate is going to discuss that in detail. And so I want to welcome Chad Mirkin from Northwestern University to talk to us about repurposing the blueprint of life. So thank you very much.
Okay, great. First, I'd like to thank the Kavli Foundation and the Norwegian Academy for this incredible honor. I have to say it's a delight to share this prize with Bob and Paul, two of the people I really like the most in science and look up to the most in science. I've developed much of my career by learning a lot from them every step of the way, and it's really great to be here, to be part of this with them. Bob talked about the importance of nanomedicine and what you can do when you begin to encapsulate drugs and control release rates and build medicines based upon those concepts. Paul talked a little bit about the importance of structure, and in the early days, when people made a set of particles, they were quite varied. Unlike conventional chemistry, where you can make a mole of molecules where all molecules are the same, you can't make a mole of nanoparticles where any 2 particles are the same when you're above a certain size. And so a lot of work's gone into learning how to refine that and use chemistry to devise ways to control that with greater and greater precision. I want to talk about the merging of those 2 concepts, because I think that's where this field's going. And I'm going to talk a little bit about where we came from, but also where we're headed as a field. And I think it's a really important step in a direction that's going to ultimately impact the way medicine is practiced for good, both in terms of how we study, track, and ultimately treat disease. And I like to refer to that as structural nanomedicine. And I want to start by kind of looking at the development of medicines over the years. And if you look back just 20 years ago, this was dominated by chemists. We tried to make molecular drugs where we could control the placement of every atom, and we did. And by doing that and understanding structure-function relationships, we could develop medicines that impacted life in a significant way. One thing you notice, though, if you track the development of drugs over time, is a move towards complexity, much more complex systems. So today, if you look at the top 10 drugs, they're no longer small molecules. They're biologics for the most part. Bob acknowledged that in his talk. Proteins, nucleic acids, for example. The top drugs, thanks to much of the work done by Moderna and Pfizer, are now nanodrugs, drugs that took advantage of this idea that you could encapsulate components like mRNA and deliver them to create very powerful medicines in the form of vaccines. And so one of the things that the drug industry's understood, and we as scientists understand, is that there's an advantage to this type of complexity. It doesn't mean that small molecules go away. They're going to be here forever, and they're going to contribute significantly into the future. But now there are lots of opportunities to build much more complex medicines that can attack disease in new ways and create treatments, in fact, for certain types of diseases that were previously undruggable. Now, here are the challenges when you think about developing drugs, and this is why it's so difficult. Bob mentioned some of them and some of the hurdles that he overcame in his talk, but there are lots of them. On the in vitro side, meaning when we're not in the body, just thinking about how they interact with cells, aggregation leads to reduced efficiency. Degradation, your body has all sorts of mechanisms to degrade drugs, so that leads to pharmaceutical lifetime. The importance of delivering multiple components. I believe many of the drugs of the future are not going to be single drug entities. They're going to have many medicinal components ultimately delivered in the form of a nanostructure that is not interesting because it's small. It's interesting because it's large. You can take and put many components within one nanostructure and present them in different ways, and that's going to be an important part of the concepts that I'm going to share with you today. That can lead ultimately to greater specificity, which is critical. You want to hit target and stay away from off-target type of effects. That leads to some of the adverse side effects that people often cite that we know are associated with almost all drugs. And then when you move to in vivo challenges, they're even greater. How do you get these drugs to the organs that ultimately matter? Once you get them to the organs that matter, how do you get them to the cells that matter? Even the compartmentalization, those are big issues. How do you avoid inflammation? How do you control the pharmacokinetics when you have many components, the signaling rates? That's really important. And then ultimately, the heterogeneity of the population. Not everybody responds the same. So these are all big challenges, and they require an understanding and a chemical design set of principles that allow you to ultimately create drugs that are efficacious. And so, as I said, if we think about this from a taxonomy standpoint, we start out with small building blocks that are the basis for most drugs. I call them unstructured components. Small molecules, DNA can be a drug, proteins, carbohydrates, RNA, and peptides all are the basis for different types of therapeutic agents, or therapeutic agents in their own right in many cases. But one of the things we've learned through the last 10 years or so is that compartmentalization in nanostructures, the vaccines is a prime example, can lead to some advantages. You can co-deliver the components that matter. You can even use the particle as an adjuvant in that case. The mRNA can be used to elicit or generate a particular protein that creates immunogenic response, but the particle can actually be an adjuvant to stimulate the immune system so the patient actually responds appropriately to that event. But one of the things that we've learned, I'm going to use an example, a prototypical example that came out of our lab, is the importance of structuring these types of elements within the context of particles. And we weren't trying to develop medicines when we first started. We were actually thinking more like a chemist. We were trying to create tiny little particles that were modified with DNA that could be the basis for programmable atoms. What we found was that when we modified these types of particles at high density, we could create constructs that had properties that were completely different from DNA, as Naomi alluded to in the introduction. And those properties turn out to be quite enabling. And so Bob talked about the central dogma. This is important. I don't think about it in the hardware terminology. I think of it as the blueprint of life, and the mRNA are the instructions, and that generates the proteins, which are the building blocks, and of course all living things are made up of these building blocks. And figuring out how you engage with each of these agents can lead to the development of new types of therapeutics. So as I said, one of the things that we did early on in our work was we began to ask the question, could we take these tiny particles, these beautiful particles people like Paul were building, and modify them with oligonucleotides? And the concept was that we could create a programmable atom. Particle, which would be our building block, and the DNA would be kind of a bonding agent. An oligonucleotide can be designed to have recognition properties. You don't have to be a rocket scientist to design it. AT, GC. We know the base pairing schemes of DNA. We could create little single strands rather than sticky balls that could recognize different targets and different particles based upon the recognition elements that we encoded on the surfaces. But when we made the first particles, we began to study them, and we discovered that they had all sorts of fantastic properties that were not just interesting in material science, but very interesting in biology. So that's kind of a common theme here, people crossing fields in terms of developing new advances in the areas of medicine, for example. And so you can see on this slide in a cartoon fashion on the left, a spherical nucleic acid made from a liposomal core with a shell of a oligonucleotide. The first particles were actually made of gold. And you can see the electron microscopy image up in the middle section here, where you can clearly see the gold core, and you can see the shell of a oligonucleotide, the halo of a oligonucleotide of DNA surrounding it. And that was kind of spectacular. So we knew we were kind of onto something, at least, that our vision of these types of structures were beginning to mirror at least the cartoons that we were drawing at the time. We began to study the properties of these structures, and as I said, we noticed some incredible differences. They would bind complementary DNA a lot more tightly than normal DNA, linear DNA, the same sequence. Well, that's important if you're thinking about developing a medical diagnostic, for example, because you can fish out a lower concentration of target than, for example, with a molecular probe based upon these types of constructs or these types of oligonucleotides. They resisted nuclease degradation. That's because they're sterically congested. Nucleases designed to degrade the oligonucleotides couldn't act on them as well. That meant they would have a longer therapeutic lifetime. They'd live longer inside living systems. That was good. They didn't elicit immune response. That was nice. And unless we designed them with oligonucleotides designed to trigger an immune response. We actually do that in the development of vaccines based upon these concepts. But the last one was really amazing. It almost took us 10 years to run this experiment because you're taught as a biologist and as a chemist, DNA does not naturally enter cells. And so we did a cell culture experiment. We just, Friday afternoon experiment, said what happens when we put DNA into a cell culture, in this case epithelial cells, make the same, use the same DNA to make a spherical nucleic acid, so exact same amount of DNA. From a particle-to-particle comparison, actually 100 times lower concentration on the right, and you can see the particles in this case, because we had dyes on them, go in remarkably well, better than anything known to scientists. And they're actively recognized by things called scavenger receptors and rapidly internalized. And you can see on the left, the same DNA does not enter cells. And at that point, we knew we had something pretty spectacular. That's kind of the aha moment in the lab where you start saying, wait, now I can, without using toxic transfection agents, move large amounts of nucleic acids into cells and these particles into cells. And that means I can begin to manipulate what goes on in cells and perhaps measure what goes on in cells. And so that sent us on an odyssey. And so the first paper was published in 1996. And again, it spanned material science and these concepts in biology. That to this day, I believe, is still the most cited paper in all fields from Nature in that year. It's pretty remarkable because it was a very different idea, right? And this concept of merging these two very disparate materials and leading to these capabilities, both in material science and biology, was actually really opening a big door. And it led us on a path where we first began to develop diagnostic tools, which I'll share with you, using these types of particles as probes to measure nucleic acid signatures in vitro, so outside of living systems, then in cells. And then we began to think about how we could use the constructs themselves as medicines to create a whole new class of medicines that are now deep into clinical trials in certain cases. And so the first example of this was developed along with my group by a company called Nanosphere that we started. And they developed a gene chip type of assay. And the basic idea was that you could have a chip that had many different spots of DNA that could interrogate for different signatures of disease. And so a sample of blood, saliva, or urine, you could process it, release the nucleic acid markers. And if the disease markers were present, they would bind to a portion of that chip and get sandwiched with a spherical nucleic acid gold nanoparticle probe. And because these are intensely colored, the idea was that you would have a really sensitive detection system. Well, we didn't know a whole lot about detection at the time, and it turned out that was partially true, but it wasn't really sensitive enough to really make people turn their heads and say this would be a good thing to do, because we were about 4 orders off in terms of sensitivity at magnitude. And then what we figured out is we could use another property of nano gold, which is not characteristic of bulk gold, and that is that it's catalytic. And we could flow photographic developing solution over this chip, which is silver 1 and hydroquinone, and anywhere where we had gold particles, we would have played out silver. And that would increase the signal by a factor of 100,000 in about 5 minutes, and now we're off to the races. We had a really sensitive assay for nucleic acid signatures disease. We could measure the presence of these silver spots by bringing light in through this is just a microscope slide with different spots of DNA, and measure the scattered light coming off these silver spots. And here you could see a typical type of panel assay. This is for HIV, Ebola virus, smallpox, and hepatitis B. And you could read with the naked eye. You know, this is done 6 times over, so all of them are present here, 1, 1, 1, 1, 2, 2, 2, 3, and 3. And you can see the background. We thought this was great. We'd have a system that you could read with the naked eye. At the time, the world wasn't ready for that. They say, no, Chad, everybody, there's people that are colorblind. So we need an instrument that could do this, and they built something called the Verigene system. This system now is in over half the world's top hospitals. So over the course of about 20 years, this has matured into a technology that is now used all over the world. Nanosphere was bought by Luminex. Luminex was bought by Diasorin, and these are used probably in hospitals that you've ultimately been at. You can see the current system shown here for bacterial infections, viral infections, cardiac tests, human genetic tests, and pharmacogenetic tests as well. Well, we didn't stop there. The basic idea here was, well, let's go further. I haven't even talked about how we take advantage of the cellular entry properties of these, but maybe we could begin to develop probes that would enter cells and allow us to measure what's occurring in cells. Small molecule content, RNA content, for example, looking at gene expression in cells. And that led to a technology called nanoflare technology. And nanoflares are simply spherical nucleic acids where you have a gold particle core. That's important. And then we take a short oligonucleotide called a flare sequence, and we hybridize it to the nanoflare construct so that the fluorophore is near the gold. And what that does, it turns off the light. That no longer fluoresces, it quenches it. And these are designed so that when they go into cells, if there is target mRNA present, that will bind, and because it's longer than the flare, it's favored thermodynamically, and it peels off the flare, releases it, and turns on the fluorescence. And so now you can begin to measure for the first time RNA content in live cells. And you can see, you can see the effect right here. These are cancer cells that overexpress a gene called survivin, so they produce large mRNA transcripts, large number of mRNA transcripts compared to cells that don't. And you can see the difference in terms of background here. Even with the naked eye, you can see the difference between these two. Now the way this is used is people will then couple this with flow cytometry and take a group of cells, maybe healthy with a few cancer cells, feed all the cells the flares, but only the cancer cells light up, and now you can fire them through a flow cytometer and separate, identify, find the needle in the haystack, but they're collected in live forms. You then can collect them and begin to look at how they behave, for example, in the presence of experimental therapeutics. So it's actually a first step towards ultimately developing drugs based upon this understanding as well. And so it's used now for isolating circulating tumor cells, high throughput drug screening, and it's even been used not by us, but a group in Singapore for in vivo imaging. In this case, they were looking at the formation of cells that were indicative of keloid formation, and there are aberrant genes that are associated with that process, and they were able to show, at least in rabbit models, that this was in fact possible. In living organisms, you could use flares to identify the presence or absence of those types of cells. We spent a lot of time not just making structures, but trying to understand how they ultimately work. And so one of the questions was, why are these being recognized and taken up? Well, it turns out the three-dimensional structure defined by a spherical nucleic acid binds very tightly to these things called scavenger receptors, which are common to most cells. That leads then to an internalization via a process called caveolin-mediated cytosis. And initially, all of these constructs go into the endosome. But then a small amount is ultimately released into the cytoplasm. That's what we ultimately use when we're measuring mRNA, for example, in the detection type of event. That's also what we're going to use if we develop a drug based upon antisense therapy, where we have to intercept mRNA, or if we want to degrade mRNA, or if we'd like to do gene editing, we have to develop constructs that can ultimately not just get to the cytoplasm, but can ultimately get to the nucleus. And so you can kind of see that this understanding then created a whole new set of thinking about how we might develop drugs that could rely on and take advantage of these fantastic properties. And so here are examples of that. We now have drugs that accelerate wound healing, diabetic wound healing, for example. So in this case, a siRNA drug that degrades a mRNA transcript and stops the production of a particular protein that inhibits this process. Psoriasis, where we again use a spherical nucleic acid to soak up RNA and to basically dial down like a rheostat the production of inflammatory proteins. Retinal diseases, macular degeneration, similar type of thing, going after VEGF. We can put these in eye drops and they'll actually function in the eye, at least of rabbits. Several of these actually have been in clinical trials, even in humans. The psoriasis drugs in particular have been taken in the early stage of clinical trials. So we're moving past small animals to humans as well. And then glioblastoma, which is a really interesting one, a deadly form of brain cancer. Not much has really been done of significance in the last 30 years in ultimately treating this disease. It's a horrible diagnosis if you know anybody that's ever had it. Typically 6 months to 18 months to live. I had a colleague who said, you know, these seem to have fantastic properties. What would happen if we put them into the circulatory system via IV injection? You know, would they cross the blood-brain barrier? And I said, you know, I have no idea, but we can try. So we did some early animal studies, and sure enough, that they do in fact cross the blood-brain barrier. He then ultimately developed a drug based upon going after a protein, mRNA, that produces a particular protein that's associated with the disease. And the idea was that if you could downregulate it, you could make these types of cancer cells susceptible to treatment with chemotherapeutics. And in animal studies, you can see that it in fact worked, and we got a significant improvement. Not a cure, significant improvement in terms of animal survival. Well, that was kind of exciting because that was the first example of a spherical nucleic acid showing properties in vivo where you have IV type delivery that looked really quite attractive in terms of treating a really debilitating disease, and a disease where it's very difficult to access the organ, the brain in this particular case. And based upon that trial, or those early studies, it led to a clinical trial. And I couldn't run this clinical trial. I had to be separated because we developed the drug from a conflict of interest standpoint. But Priya Kamthekar and Alex Stagg ran this trial at Northwestern. And I just want to show you the video from it, because to me it's really quite interesting. It shows the importance of, you know, this early type of work and understanding just how the drug functions that ultimately leads to the development of more impactful drugs. So I'm going to play a little video here where they describe the trial to some extent.
Breaking through the blood-brain barrier. In the lab, nanoparticle spheres packed with RNA, poised to deliver a deadly message to cancer cells, moved freely through the bloodstream, crossing the protective blood-brain barrier and penetrating tumor cells, ultimately sparking cell death.
When we look at them in a lab setting, we can see a lot of impact to glioblastoma and glioblastoma cells, but it's a totally different game when we're talking about getting it to the brain tumor of our patients.
Doctors hoped the same thing would happen in humans.
There hasn't been a new drug approval for glioblastoma in over a decade. And there's a lot of different reasons that make this tumor so challenging to treat.
To test out the nanoparticle and determine its safety, researchers enrolled 8 glioblastoma patients in a small study. Tahara Sanders was among them.
You don't want to have this disease and just have it, and that's it. It makes you want to do your part.
We spoke with Tahara in 2017 after she received an infusion of the special nanoparticles.
It probably is post-surgically related.
24 hours later, she underwent surgery to remove her glioma, and doctors looked for signs the nanoparticles made it to her tumor.
I have some faith in it, I really do.
4 years later, the results are in. The nanoparticles not only made it to Tahara's tumor, but the compound triggered the death of cancer cells. The same result occurred in all participants tested post-operatively.
Yeah, so this is an example. I mean, first of all, those people are amazing that they do this, because they sign up for these trials. There's very little chance that it's going to have a significant impact, especially for a drug like this, first time it's tested. But what you get out of that trial was so important, and has actually fed now the development of subsequent drugs. We know that they're safe in terms of injecting. That was not true. Nobody knew back then whether this was going to be the case. 2 is we know they get to the brain. 3 is that we get to the tumor, and then 4, that they actually hit target, because in all these cases the tumors had to be removed and we could actually look at them. And so in many respects, that helped pioneer the development of some of the subsequent drugs. We then began to think about, what is the real strength? How can we really go for the low-hanging fruit here? And we turned to vaccines because if you understand the pathway of uptake, we know that 100% of the drug at some time or another is in endosomes. And we began to ask the question, what types of drugs could we develop that would begin to target targets that resided in the endosome? And there are things called TLRs, toll-like receptors, that are associated with elevating your immune system if engaged properly. And there are different DNA sequences known to do that. So we could create spherical nucleic acids that in principle could be great adjuvants. And so just for those of you that aren't familiar with how vaccines work, vaccines are basically 2 components. You have to have something that stimulates the immune system and something that trains the immune system. A stimulator is the adjuvant, a trainer is a peptide. In the case of an mRNA vaccine, you're putting an RNA signature in there that generates the particular protein or peptide that ultimately stimulates the immune system. But those are the 2 components. And so typically, if you're going to deliver these as discrete entities, you want to get them into what are called dendritic cells. And then this is really important. These are processed and put on the surface of the cell in the form of the antigen. The antigen's taken, and that's going to give you the selectivity that we're talking about. And there are these other things called co-stimulatory markers that are generated as well. And I point to that because the rate at which this occurs is really quite important, but very little attention is paid to controlling that ultimately. And that's where structural nanomedicine is going to play a big role. Once this happens, T cells dock, and that's the training mechanism. And now they then go out through the immune system and ultimately find cancer cells. And if the signature that they've been designed to recognize is present, unique to a cancer cell, they lyse those cancer cells, and the cells die, and the tumor presumably or hopefully goes away. And so I like to think of the current way we develop vaccines as the blender approach. It's completely different from the way I opened up this talk, where chemists spend all this time controlling structure. Here, all they care about is the adjuvant and the antigen. Mix them. If they work, run with it. If not, discard. The development of the mRNA vaccines, a major step in the right direction. At least that's saying, hey, compartmentalization is going to be important, and delivering these things as one entity and in a controlled way is going to lead to some advantages. That's been proven. But I think there's a new possibility, and that is using structures as modular entities where we not only can deliver the components, but control the placement of them in a way that maximizes efficacy. So distilled down to the simplest terms, this basically says structure matters. That's not shocking to a chemist, but when you talk to a lot of medical doctors, no offense, some engineers, they discarded it initially, right? But I think they're going to change their tune after you start seeing some of these results. So here's a first example of structure mattering. So these are Merkel cell carcinoma patients. And we went through an early trial where we began to ask, could spherical nucleic acids made of immunostimulatory DNA be used as adjuvants, immune stimulators, in a therapeutic trial? And these patients all failed treatment with all existing options, including things called checkpoint inhibitors. You hear about them, Keytruda, Pembro, those types of drugs. And so they sign up for trials because they're hoping that maybe these experimental trials will make a difference. In this case, the idea was we could jack up a patient's immune system with a spherical nucleic acid in combination with a checkpoint inhibitor, in this case Pembro. This is a 92-year-old patient before and after treatment with the drug. This person is alive because of this type of treatment. Pretty remarkable. Now, not all the patients responded in this way. I think 3 out of 14. But a sign we're moving in the right direction. And that's kind of set us off in the races. And we think this is actually going to be a modular platform to develop a whole lot of important drugs going forward. But that's not a vaccine yet. To get a vaccine, you somehow have to introduce the concept of an antigen into these structures. And we can do that pretty easily because if we build these out of liposomal type architectures, we can put, for example, peptides or RNA in the core. We've done both. I'm showing you the peptide example. Oligonucleotides on the outside that are designed, for example, to be adjuvants. So we're going to compare this to what I call the blender approach, right? The stimulant here and the antigen. Antigen is in the core here, or we synthesize an antigen with a tail that can insert itself into the liposome, be interspersed with the adjuvant DNA, or we synthesize peptides that have tails of DNA They can hybridize to the adjuvant DNA, and that keeps them together. So all three of these are actually compositionally nearly identical, but they're clearly structurally different. And so this gave us a chance to test that hypothesis that ultimately structure matters. And you can see in the trial here, in this case a small animal trial, this is for HPV. These are for the aficionados, the DNA and the peptide used, the antigen and adjuvant. In the absence of anything, a tumor grows and animals die. You can see over here in the survival curves. If we use the mixture, there's almost no effect under these conditions. If I create the encapsulated structure, that was really disappointing when we got the results. That had almost no effect. You can see the green here, very modest effect, if at all, in terms of increasing animal survival. You go to the anchored, though, this is starting to get interesting. Now you're really suppressing tumor growth. You can see that in the animals surviving. And then you get to the hybridized structure, and it's almost a complete cure. Okay, so this was where I thought we are really onto something. This idea of structure matter, could be really quite consequential here. This is where I truly believed it was the case. We then went through 9 different models. You can see the different types of cancers on here. And I just want to point to a comparison, in this case what we call the privileged architecture. This has the hybridized entity. Different antigens, different types of diseases. In every single case, you move down the ineffective to curative axis. You don't get cure in all cases, but you always get better. And that's pretty profound if you think about it, because it means, one, this might be a great way of developing medicines, but two is we should go back and begin to look at some of the antigen-adjuvant combinations that we passed on. It might not be because they were the wrong adjuvant antigen, but just the wrong structure. And we've actually taken 2 cases, including ones that failed clinical trials, and shown that is in fact the case. And so I think this is going to be a concept that's ultimately is not just in spherical nucleic acids, but across the board. And that's why I'm so bullish on it. And so if you just kind of ask, how good are these for, again, the aficionados, 80 times increase in APC activation, antigen-presenting cell, 35 times more APCs receive both components. That's because you're putting them together. A 15 times increase in antigen presentation. These are all the things that matter in developing a vaccine. And 6.5 times increase in tumor T-cell tumor killing. Okay, that's kind of going and compartmentalizing and beginning to look at crude structural control. As I said, one of the things that we're trying to do is really move towards building molecularly pure architectures. And we might ask, well, how would that might manifest itself, and where would there be some significant advantages? Well, it turns out I was one of the lead experts in defending the CRISPR patents on the Broad side, the side that won, by the way. And learned just a tremendous amount about all the issues involved in gene editing. To me, a remarkable tool, by the way, worthy of Kavli Prize, worthy of a Nobel Prize. I think one of the most important nanomachines ever discovered. But incredible that it even works, because you need multiple components coming together in a cell, avoiding all the compartmentalization, getting to the nucleus, latching onto the target in the right place, and affecting gene editing. Yet these are all generated at different parts in the cell, and somehow it ultimately works. We began to think about this in a different way. We said maybe we could begin to use this concept of spherical nucleic acids and transform the proteins that are part of the gene editing machine into spherical nucleic acids, preload them with the right type of guide RNA, and then deliver that as one construct where everything would be together. And if it was a spherical nucleic acid, we'd overcome one of the biggest barriers in this field, which is cellular and tissue access. And so the idea was create CRISPR SNAs. And so just to make a long story short, we've actually done that. We can make Cas9, and we can make it in specialty forms. This is the protein used in gene editing. On the ends, in the N-terminal side of the protein, we can put peptides on them. That's important because that can facilitate the escape from endosomes, because we have to get to the nucleus. We don't want to be in the endosomes. On the other side, in the C1, where is it? Should be an NLS sequence here. Oh, right here. Yeah, we can put another peptide that actually guides this machine to the nucleus. We can put a shell of a oligonucleotide on the Cas9. That's going to get us into cells and get us into tissues that ultimately matter. And we can preload the guide RNA and basically take it to specific targets of interest. And then we can ask, ultimately, does it work, and how does it work compared to the conventional CRISPR cases? And it turns out this, in fact, works quite well. No big surprise, by making it a spherical nucleic acid, we get that scavenger receptor-mediated endocytosis, and it's significantly increased, so that's important in terms of efficiency. We can track where the cassette is, where the construct is, using labels, and we can traverse from the endosome to the cytosol, ultimately to the nucleus. And this is really where the rubber hits the road. We can ask, will it snip DNA in the right place? And we get very efficient gene editing, even in very difficult to edit cells. So here you can see across 3 different cell lines and the degree of gene editing that we're seeing. So I think this is important because this is a step towards where I said we want to go. Remember, I said we started out with really making medicines where they're chemically well defined. We then go to average type structures, including the SNA vaccines that I shared with you. This is now moving towards molecularly precise architectures. We just wrote a perspective article in Nature that will hopefully be coming out in a few months. I took this from that article, and it really highlights that. These are the components that we typically deal with respect to developing medicines. We're kind of heavy into this particular stage where we're beginning to create these larger constructs, these average, from a definition standpoint, type constructs that are showing real important efficacy advances and advancing capabilities. But I really think this is the frontier. How do you begin to use a lot of the precision Paul was talking about, put it all together and begin to make constructs that have been designed to do the things you want and to traverse all the barriers that we've talked about to become the most potent medicines with the least side effects. And we'll see. That story is being told, and we're going to find out how far we get in the next couple of decades, but it's going to be an exciting ride. Let me just finish then by acknowledging the people that have funded the work, the people that have helped develop the work. This is for my 60th birthday, Paul. Well, this is half the crew. They're all over the world as well. But it was a pretty exciting one to see them all back. It is an enormous team of people. I put this up here because I can't go through all their names, but just an enormous team of people that have contributed. Graduate students, postdocs. My daughter, Sarah's in the audience. She actually has a patent on a spherical nucleic acid. Silica, of course. She asked, what about the royalties? Unfortunately, not producing many royalties. And then just incredible colleagues from all over the world in Northwestern. I've got great people. We have a wonderful team. We have a saying at Northwestern, we like to hunt in packs. And I always say my pack is pretty darn good. We've been able to grab whoever we needed at the medical school and engineering to really do the things that we want and to move into new areas where, frankly, we weren't originally trained. And that's been a lot of fun as well. And then I just thank my wife, who's here as well. Beth has been very important in terms of helping us get to where we are today, been incredibly supportive in everything that we've ultimately done. And then I want to finish with give me 20 seconds, because I violated a rule today. I used to think early in my career when I'd go to these award ceremonies and we get these starter grants and they're honoring everybody, never go up against a paleontologist or an astrophysicist. And I really learned that rule was true, and I violated it today because at breakfast this morning, my brother Andy said, do you think it'd be a bad form if I skipped your talk and went to the astrophysics talk? So I'll stop with that, and thank you guys all for paying attention.
Thank you very much, Ted. Do we have any questions for Ted? So, yes.
Thank you. Great talk. I heard you talk once about maybe some lack of encouragement you got early in your career that helped lead to your persistence in pursuing science. And I thought you might share that with some people who struggle with things that Paul and Bob have talked about when people say this isn't possible or we don't believe in you. I think it was a letter you got at Northwestern after
Oh yeah, it was great. No, so look, I have many experiences. I think everybody would say this. Maybe things have changed a little bit, probably for the better to a lot of extent. But it was pretty brutal early on. I mean, science was a contact sport, so to speak, and people weren't always the kindest, especially when you were a new person coming into a field. And I was struggling. I was doing really well getting these honorific type things because we had a really good record as a postdoc, and that's what you get these honorific things for. But breaking into the general funding system was not easy. And I remember my 4th year, I got a review from the National Science Foundation, and it kind of went something like this. You know, Chad Mirkin's been at Northwestern for 4 years. He's done nothing of import. I see no reason why the federal government should waste another dollar on his research program. And I took that and I posted it outside my wall, and I wanted every student and every postdoc to see that, because, you know, you have got to fight through it all. I mean, you know, if you truly believe in what you're doing, you should always try to learn from what people are saying. But if you believe, like Bob was saying, you got to go with it. And if you have conviction, you know, sometimes Lise just banging your head on the wall, but other times you step around or you go through it, and good things ultimately happen. So if you truly believe in it, I think you need to keep pursuing it. I mean, put another way, I was once a commencement speaker that was giving everybody a pep talk about, you know, going on from Northwestern in life, and it's a guy named Michael Wilbon. I'll give him credit. He's a big sports guy, and he said, you know, you've been told your entire life how to succeed. You've been told to have a plan, and all you guys are fantastic as you're graduating from Northwestern. I'm sure you've all had a plan. He goes, but I'm going to tell you, or I'm going to quote a great philosopher named Mike Tyson, who once said, a plan's great until the first time you get punched in the face. And what happens after that is ultimately what defines what you do. And I think how you react, I think that's probably the most important lesson. You know, everybody has this happen, and you've got to fight through it if you truly believe in what you're doing. And eventually, you know, people come around and believe. I mean, I still to this day get bad reviews, and you know, I try to learn from them, but I don't go crawl in a corner.
Very good. So do you still have the microphone? Yeah, very good, very good. Any further questions? Yes. Hold on.
Your core nanoparticles, that material, when you treat people in the brain for the brain disease, once the core degradation, what happens?
Well, so we don't know in humans. We know in animals that it's ultimately excreted through the bile system. But in the brain case, the only drug that we've ever taken in the clinic that had gold was the brain drug. And we did that for a reason. The gold was going to be used as a diagnostic. You don't have much gold background in you. So when we got the tumor, we could actually measure the amount of gold that got to the tumor. And so we used it as both the basis of a therapeutic, meaning the shell of a oligonucleotide. In that case, it was an siRNA designed to target BCL2L12, which is the gene I was referring to. We could then measure the amount of the gold using ICP mass spec, quantify it, and it was designed for that purpose. Going forward, all the drugs will have liposomal cores because those are biologically benign and ultimately just go into the background. You've got lipids and all sorts of other things that are very similar to them, and that's a much more attractive basis for drugs going forward.
Yes.
I have a question concerning all your targeting signals that you had for your nanoparticles, your circular RNA going into the brain. And then there was one thing missing. That was the final step of actually getting to the loading compartment of the antigen. Do you also put that into your RNA when you produce the antigen?
No. The antigen in our case, in the case of the vaccines, is actually a pre-synthesized peptide. That is either put in the core, that was the internalized structure that I showed you, the encapsulated structure, or we synthesize a tail that allows us to insert it into the liposome. We terminate it with a cholesterol or tocopherol group, or we synthesize an oligonucleotide tail on it and hybridize it to the adjuvant DNA. So those are pre-made peptides that we're using as antigens. We actually have versions where we carry the mRNA inside the core and then get the cells to produce the antigen of interest. But we wanted to look at both scenarios. I shared with you the data from the pre-made antigens.
So I have a question. So with more and more of these more complex structures where we have DNA or RNA, we also have an opportunity for intercalating small molecules. So even though things have moved away from small molecules therapeutically, you've created some new opportunities for delivering or dosing like neurotransmitter, metabolites, different sorts of molecules that maybe the study sections have kind of moved away from, but actually you have some significant...
No, I agree. All that's coming back. So again, everything in here, both on the nano side, but also the components that are drugs, will go into these structural nanomedicines. So, it isn't been there, done that, we're done with those. I believe many of the medicines of the future will be complex aggregates of these materials that allow you to go after diseases in unique ways. And intercalators are also examples as well, and we've actually delivered them using these types of constructs as well. So it's a good point.
So on that same side, since we're sort of thinking a little bit about intercalants, there are also a lot of toxins. We've looked at PAHs, there's microplastics, we have a lot of toxins in the body that people are also saying more than 50% of adult onset diseases might have some origin on the toxins that we're exposed to. Are there any thoughts about scavenging in that sense?
Well, there are many. We don't do a lot of that, but I'll go back to what Bob was talking last night about. You have to get clever in terms of dealing with agencies. I probably shouldn't say this because it's recorded, but I was once working with the Air Force, and you'd say the Air Force, they can't be possibly interested in this, and I said that to them. I said, we've made stuff called synthetic HDL. We've made a nanostructure that acts like HDL, the good cholesterol. And the idea was that we could use it to elevate a person's HDL level, and that could perhaps be very useful in treating cardiovascular disease. But I need to get some funding for this, and I just don't know, I can't think of a route to do it. He goes, you're not thinking creatively enough. He said, you need to think of this as a mimic to understand how you remove toxins, because cholesterol is a toxin, and if you talk about it as a toxin removal system and developing a construct that would go in and take out cholesterol, then you have a fundable idea.
There you go.
And that led to a nice grant and a lot of good studies that came out of that. So there's a lot of interest in moving in that area. We don't do a lot of that. We've got enough on our plate with these, but I think there are lots of opportunities for young people to really push the frontiers.
Well, that's a really great message also for the younger people in the audience who are thinking about a future in this area. But what I'd like to do is really conclude today and thank our 3 laureates who have really opened the door for us, or seeing what tremendous potential we have for nanoscience and biologics and for the future of nanomedicine. So please join me in thanking our